begin. So I'm just going to hit the record button, and I hope it works. All right, let's begin. Today's discussion is really centered on the subject of translational glycobiology. What is translational glycobiology, and why are we getting out of bed every morning, rushing to come into the laboratory to do the work that we do? So the title, as you see it, is sialylated Lewis X. It's abbreviated term is SLEX. It's got a CD nomenclature. It's CD15S, and the translational glycobiology of leukemogenesis. And leukemogenesis is the process, incidentally, that's misspelled. There should be an E in here, is the process by which leukemia evolves. And so its role in driving the pathobiology of hematopoiesis when it decides it's going to go down the route of making leukemia cells rather than normal blood cells. Now, to advance this, I'm a little bit unsure because I have to come to the other screen here. Do y'all see this?
Yep.
So this is kind of a rhetorical question, but if you look at this for a moment, you're going to understand a little bit more about why we are doing what we're doing. And that big panther that's outside of our door at the TGIF is really sincere. We're here to cure disease. So here's the question. If you were ill, would you rather be cured or would you rather undergo lifelong treatment? And if that doesn't catch your attention, how about this question? If someone you love gets sick, would you pray, hope that they be cured, or would you pray, hope that they undergo lifelong treatment? I think all of us would be praying for cure in every situation. Now, cure is a very interesting word because the pharmaceutical companies believe cure is crafted by treatment, and treatment can lead to life extension, but it also leads to an economic model that requires that you be tagged to that pharmaceutical approach for the rest of your life. And we will discuss today a possible drug approach towards the treatment of leukemia, but our intention is to make the leukemic easier to eradicate. So conceptually, we're seeking to cure in every case that we do research, at least in my laboratory, if not in every laboratory of the TGIF. So I bring to your attention an article that was written in May of 2010. It was actually a double issue of Newsweek. That was one of the major weekly print magazines in that era. These things have kind of gone by the wayside a bit with the ability to get everything online. But Newsweek and Time were really the two major news magazines in the United States, with The Economist being the one in England. And at that moment, the reporter for Newsweek had approached me, Sharon Begley, and asked me to comment about some difficulties I was getting in enabling a mesenchymal stem cell that would otherwise be useful for cure of osteoporosis, enabling that technology to get through the U.S. Patent and Trademark Office. And when I told her everything that I was going through, she decided to write a story about how difficult it is to actually achieve cure and how many different hurdles people face from, first of all, conception to enablement, then enablement to patent, then patent to development and preclinical models, and after preclinical models, whether that will go on to drug development and pharmaceutical stage kind of processes. And in there's this thing called the Valley of Death, where you have an idea and you may have preclinical evidence that the idea may actually be useful to solve a problem in patient care, but there's no funding to take it further. So with all of these steps, she basically dissected all of the reasons for why it is very rare for laboratory investigations to actually get to the bedside. And I really bring this to your attention because it is said that roughly 3 out of 1,000 potential druggable targets actually get to a pharmaceutical product. Very interesting situation. I think everyone should read it. So if you need a copy of this article, let me know. I can send it to you. So in the background of all this, you have to understand what are the basic types of research one can perform, because you're at a university. You're not in solely a medical school dedicated to finding answers to improve human treatment and human condition. You're looking at a place where there's a lot of different scientists doing a lot of different things. And so you have to make a distinguished thought about this and recognize there's two broad types of research. Number one is pure research, where one might have an interesting curiosity-driven question, like, for example, why does the butterfly have that color in particular terms in the wing? And then the other one's applied research, where you see a situation where you need to solve a problem and you engage to identify the processes that are underlying the problem and then try to overcome where the problems exist in the process. So pure research is the research that's generally funded by the National Science Foundation, especially in terms of biochemical research, whereas applied research, when it comes to health, is the purview of the National Institutes of Health. And in some cases, when you look at grant sources, you can kind of figure out whether the inspiration for the research was more towards delivery and real health implications versus pure, that is, unapplied science in its purest sense. Okay? So here's a cartoon taken from an article where someone tried to summarize the differences between pure and applied science. And so here's a picture of pure research scientist perspective, and the reporter's asking this research scientist, here's how this person depicts a pure research scientist. Has a marijuana joint in his mouth and beard. He looks like a bohemian, right? Pretty frail looking bohemian at that. So the question is, why is it important that we have trapped antimatter? And the pure scientist says, because it's awesome. No other answer, just because it's awesome. So curious, inspired by something that person really thought they wanted to learn more about, is pretty great. And then here's the perspective of the applied scientist. Why is it important that we have trapped antimatter? Same question. Well, let me just show you the difference in depiction. Here's a well-dressed person, maybe wearing a leisure suit, but has some kind of a sport jacket on. Certainly is not smoking some weed or whatever that thing was in the person's mouth, and seems to be clean cut. Okay, well, it has future applications of propulsion, energy creation, data transmission, you name it. Now, obviously, the person who wrote this article favors applied science over pure science. But the bottom line is that the applied scientist is looking at how it should be used as the inspiration for what it is that's going to be discovered. So when we come to this concept, which is important for all of us to understand, what is translational research? Well, translational research in the purest sense is a clinical effort. It's a form of applied research as opposed to pure research, wherein the specific goal is to alleviate human suffering. So there's your clinical inspiration. By hypothesis-driven basic science inquiry, providing mechanistic insights applicable to the patient. Now, I emphasize mechanistic because this is not descriptive science. We're not simply describing a phenomenon and walking away from it. We're observing something uniquely and then wanting to get to the molecular basis of it. But the important point is it's applicable to the patient. And ideally, it's an inspired process, inspired by medical necessity. So let's take a look at a moment of how research models are employed, because unfortunately, all too frequently, we presume that the mouse is the representation of human biology, and that is just not true. And especially in glycobiology, where mouse and other types of mammals differ significantly from human, in terms of the glycosyl transferases and the glycosidases that exist in the human genome versus the mouse genome. Now, let me stop for a second here and just tell you something very fundamental. When I was a medical student, and even earlier when I was just an undergraduate, we were starting to identify the number of genes that were present in lower organisms. And people were amazed to see that a yeast has 6,000 different genes. And then people that were studying fruit flies found that the fruit fly had 20,000 genes. And so in that era, I was instructed that the human probably has 1 million genes, maybe more, 2 million genes. I mean, those were numbers that were thrown at me all the time. And then the most interesting thing happened at the end of the Human Genome Project, when we finally understood the entire complexity of the human genome, we only have 25,000 genes. A fruit fly has 20,000 genes. If we have so much higher complexity than a fruit fly, what is it that's landing us this complexity? Well, it's the post-translational modifications of the proteins. And in large part, that's dictated by the glycans. So when you want to study glycobiology, you've got to start looking at the actual organism that you seek to find the answer in. And to get to the proximate reason for study, if it's a human, then you have to go to the proximate subject, which is the human. So I caution everybody, when feasible, possible, always study human cells to elucidate human biology. And nothing could be more critical in this message than glycobiology. Another point I want to emphasize to everybody is people make the mistake all too frequently to believe that what they're seeing in a tissue culture environment or at the lab bench is equivalent to what's happening physiologically in vivo. No, not at all. What one should try to do is obtain primary human cells from human beings as much as possible. If you have to use a cell line as a model, be aware that it's a cell line. But at the end of the day, you want to be confident that there is a model that will allow you to have the power to deduce what could possibly be happening in a human. For the case of leukemia, there are models that we employ. We can take immunodeficient mice and transfer the leukemic cells of a human and any other cancer cell, for that matter, of a human into a mouse that will propagate that tumor, that cancer, in whatever form, in a mammalian host. Now, again, there are differences in the glycobiology of that host in regards to what the donor cells, the cancer cell or the leukemic cell from a human, but nonetheless, it allows for propagation in a physiologic host. And somebody needs to mute their background, if you would be so kind. Somebody's on the, I can hear a conversation independently. Please mute. I'm going to host. I don't know if we can. Is that Erasmo? Please. Okay. Otherwise,
I was able to mute it.
You can mute him. Okay, super. Thanks so much. So let's talk about glycoscientists. There's two major camps, and quite honestly, they don't talk seamlessly among each other. Okay. There's a glycobiologist, and then there's the glycochemists. And the glycochemists really believe that they are the one true glycoscientists. But the reason for that is a lot of people are exploiting glycans insofar as they're really not embedded in the field of glycoscience. They're merely identifying how glycans affect the biology that they're interested in. So, for example, there are cell biologists that will study how carbohydrates affect cell processes, but they're really interested in the cell process and the carbohydrates and almost, oh, by the way. And there's immunologists that are studying immune cell interactions, but have recognized that glycans play a critical role in the immune reaction. So therefore are studying glycans. And for that reason, they are in titular form glycobiologists, but they're really more immunologists and they're cell biologists. But when you're a glycochemist, you're pretty embedded, full hook and sinker, right into the field of glycoscience. So there's been always this little bit of tension between glycobiologists and glycochemists. But let me put this out for you so you think about it in terms of what we're doing in the Translational Glycobiology Institute at FIU. If it's glycoscience-based research that's inspired by overarching medical necessity, then that research is translational glycobiology. Now, that can be approached from the standpoint of a clinician who recognizes a medical need, a glycochemist that's making a compound that that person may perceive could be used in a clinical setting, or a glycobiologist, an immunologist, a cancer biologist, a microbiologist, a cell biologist who understands how sugars, how glycans intersect the biology that one's seeking to identify to cure a medical problem. So understand at the end of the day, there's this Venn diagram that comes here, center on medical necessity. Smack in the middle of this is where there's translational glycobiology. I'm going to bring to your attention now a wonderful 1-hour talk by my colleague, Carolyn Bertozzi. And I'm giving you the YouTube link here. And if you have an hour, this is really worth your time. And interestingly, Dr. Bertozzi in this talk also makes reference to the conundrum of the total number of genes in the Homo sapiens being only 25,000, where there's 20,000 genes in the fruit fly. What makes up the difference? The carbohydrate modifications of the proteins that are produced by the genome. And we talk about the entire complex of carbohydrates in a given host or a given cell as the glycome, as we talk about the genes in forms of the genome. But the genome is relatively easy to put your fingers around because there's only 25,000 genes. But even though we only have 9 major building block monosaccharides in the human, the complexity of carbohydrate structure is astronomical. So that's what you need to appreciate. And that is the distinguishing characteristic, the heterogeneity of the carbohydrate contributions that change the biology of the underlying protein and or lipid, and therefore confer added biological capability on the same scaffold molecule, a protein or a lipid. So Dr. Bertozzi, just a year ago, wrote a very comprehensive review on the subject of glycobiology and looking at 3 major classes of molecules within the field of glycobiology, the selectin, the cyclics, and the galectins, as models for how glycobiology offers important opportunities to cure disease in humans. And there's 2 figures in that paper I want to bring to your attention. Figure 1, the very first figure, she talks about the selectin class of adhesion molecules, E, L, and P-selectin. And as most of you know, all the selectins bind a similar carbohydrate motif, sialylated Lewis X, or CD15S, SLX. And E-selectin is the endothelial selectin, whereas L-selectin is found on leukocytes, and P-selectin is found on platelets, but can also be found on endothelial cells. So P-selectin can be placed on both a platelet surface as well as an endothelial surface. But she points out the importance of E and L and P-selectin ligands and highlights most prominently this molecule H-cell. She also makes a connection here between E-selectin and L-selectin because some people have obtained evidence that L-selectin can function as an E-selectin ligand. We've never seen that in our laboratory despite decades and decades and decades of work on this. I mean, 3 decades we've worked in this area. We've never seen L-selectin support E-selectin binding, but in some laboratories under some circumstances, which we've never been able to, unfortunately, duplicate, people have observed that L-selectin may be decorated with sialylated Lewis X and function as an E-selectin ligand. But in general terms, the principal E-selectin ligands are shown here. H-cell, which is a glycoform of CD44, we'll talk more about in a minute. CD43E, a glycoform of CD43 that is heavily decorated with sialylated Lewis X, and we call it CD43E, actually to identify that specific glycoform of CD43, and a glycoform of PSGL1, which should have been called CLA here, because PSGL1 functions as a P-selectin ligand, but if it's more heavily decorated with sialylated Lewis X, it can serve as both a P and E-selectin ligand. But since it was first identified as a P-selectin ligand, and subsequently was identified if it's more heavily decorated with sialylated Lewis X to be an E-selectin ligand, we call this glycoform of PSGL1 that binds E-selectin CLA for cutaneous lymphocyte antigen. So in figure 7 of this same important report, I want to bring out 2 things which should catch your attention. Look at the timeline of key developments in translational glycobiology. Up here, the discovery of H-cell in 2000, and in 2008, the identification of H-cell as a principal mediator of homing of hematopoietic stem cells into the marrow. And in 2005, the observation that leukemias home to E-selectin positive regions of the bone marrow. And this is true in mouse and is in humans. So we come back to this business. If you want to study leukemogenesis in a human, get access to human cells. Well, thankfully, because I'm a bone marrow transplant physician, and we have another one on this call today, Dr. Mark Murakami, we can get access to bone marrow cells and actually isolate normal or native human hematopoietic progenitor cells. We can also gain access to leukemic cells from a bone marrow from a patient with leukemia merely by doing a bone marrow aspirate. And so one can obtain primary cells in real time immediately that can be observed to function in the way they would have in the human in vivo by using that opportunity to grab those cells and study them immediately after they've been obtained from a patient. We have a number of different markers that allow us to identify the authentic human hematopoietic stem cell, a pentat of markers that include this thing called CD34, which is a principal marker of all human hematopoietic progenitor and stem cells. But CD34 expression extends to a variety of different lineage-specific stages of human hematopoietic cell development. So it's not a pure marker of the hematopoietic stem cell, but identifies instead the earliest progenitor population in the hierarchy of hematopoiesis. But if you come in with this group that's CD38 negative, CD90 positive, CD45RA negative, and CD49F positive, this 5-marker combination will pull out the authentic human hematopoietic stem cell. And then there's a variety of different stages that can be identified by presence or absence of these markers in a variety of different combinations. And then you get to this common myeloid progenitor, which will make your granulocytes, your neutrophils, your basophils, your eosinophils. They'll also make monocytes. And then over here, it'll break off and make megakaryocytes, which will make platelets, and erythroblasts, which will make red cells. And then over here is the mixed lymphoid progenitor, which will make lymphoid cells and some forms of dendritic cells as well. Incidentally, dendritic cells can also come off of this lineage. If you were to look at literature that was created by the pharmaceutical industry in the late 1990s, you would believe that in order to achieve hematopoiesis, all you have to do is put certain cytokines in a soluble condition in kinetic sequence, and you would be able to drive the elaboration of hematopoietic cells and final formed elements simply by introducing different cytokines at different times and in different combinations. That type of logic was promulgated greatly by the pharmaceutical industry because they were making cytokines that could be tweaking human recovery of blood formation after a bone marrow transplant or in patients with aplastic anemia and so on and so forth. And so they felt that this was the way to make it simplistic idea that cytokines drive human hematopoietic development. Well, they do, but not just cytokines. More importantly, cell-cell and cell matrix interactions. So rather than looking at this as something that can happen in a test tube, you really have to understand that it happens on a solid support environment that is the marrow. And this is fat. This is marrow fat. Marrow has bucket loads of fat. It actually tracks in percentage of marrow population, your age. So when you're 10 years old, you only have 10% marrow fat. When you get to 90 years of age, you have 90% marrow fat. Here's a megakaryocyte, incidentally, here in the middle, that all of these are differentiating stages of red cells and neutrophil series cells, all embedded in an appropriate microenvironment that allows the growth of the cells. And then for the case of the most primitive cells, the hematopoietic stem cells, we talk about the hematopoietic niche, an area which is uniquely created to support the earliest hematopoietic stem cells in their ability to continue to be the mother cell, that is, do self-duplication, plus then differentiate towards progeny cells. That's a unique niche that allows a cell to do one of two things concurrently. Well, we got to get back to the subject of these selectins, which I brought to your attention at the first couple of slides with the interest that Dr. Bertozzi had put forward in her seminal review just last year. So let's talk about this molecule E-selectin, CD62E. It's got a CD62 family nomenclature, and E is E-selectin, L, CD62L is L-selectin, and CD62P is P-selectin. Now we're going to talk about CD62E, E-selectin. This is a cell surface protein that belongs to a family of proteins. The whole family is called selectin, CD62 family, that bind to sugar structures, i.e. there are lectins. Embedded in the name, in fact, is the word lectin. A lectin is a protein, by definition, that binds to a carbohydrate. So most of you have learned protein-protein interactions and not really considered how proteins can bind to other structures. Well, importantly, proteins do bind to sugars, and that biology is critical to every process in a human. Those are the lectins, the proteins that can bind to the sugars, and selectins are a family of lectins. So in this case, selectins bind to a defined structure of glycans, a tetrasaccharide motif that we know as sialylated Lewis X or its isomer sialylated Lewis A that can be presented on a lipid scaffold, in case that it happens. That's a glycolipid, or on a protein scaffold, in which that happening is called a glycoprotein. Now, E-selectin is normally found lining the vessels of bone marrow and skin, importantly, the microvessels of bone marrow and skin. But it's induced and highly expressed on vessels at all sites of tissue injury induced by injury itself, any traumatic injury, burn, ischemia, any of those sorts of things, but also markedly upregulated by TNF and IL-1. Markedly upregulated. Now, take a look at this TNF, tumor necrosis factor. That cytokine was first isolated from solid tumors, and tumor necrosis factor is found in highest amount in cancer tissue. So therefore, E-selectin is markedly upregulated in endothelial beds in cancer. Now, E-selectin functions to capture cells and blood flow as they happen to flow by in hemodynamic shear. And also importantly, E-selectin receptor ligand interactions are critical to hematopoiesis. So let's go into some detail about this. What is this thing, sialylated Lewis X? Well, here it is, a structure of 4 monosaccharides in distinct linkages comprising sialic acid, here, neurominic acid, galactose, and acetylglucosamine, and then out here in the breeze, alpha-1,3 link, we call this an alpha-1,3 linkage, regio and stereo specific linkage between fucose and acetylglucosamine. So every one of these linkages is critical, and I don't want to get you caught up in the organic chemistry here, but this alpha-2,3 linkage is critical for the sialic acid on galactose. In this case, for sialylated Lewis X, the beta-1,4 linkage between the galactose and the N-acetylglucosamine. And again, for the case of sialylated Lewis X, fucose alpha-1,3 link to the N-acetylglucosamine. Then over here, instead of this R, we could have it, instead of this H rather, we could have an R, which be a continuation of this carbohydrate on other sugars. And the usual sugars that carry this terminal motif is a lactosamine chain. So we generally see this as a polylactosamine terminus sialylated Lewis X. We have a way of depicting this in cartoon and more simplistic fashion rather than putting up these Fisher nomenclature diagrams. We just use a purple triangle denotes sialic acid, otherwise called neurominic acid, same terminology for the same monosaccharide. Neurominic acid is sialic acid. Galactose is always a yellow circle, and acetylglucosamine is always a blue box, and fucose is a red triangle. So a cell in flow that is decorated with sialylated Lewis X or its cousin, the isomer sialylated Lewis X, or any other combination of either one of them, will be looking for E-selectin expressed on the endothelium of a blood vessel in a target tissue. And if it finds it, it's going to land on this thing. And when it lands on it, it'll begin the process of tethering and rolling, ultimately leading to transmigration, where the cell will exit the vasculature. And I'm going to go into a little more detail about that in a moment. I want to point out what sialylated Lewis X is. It's an isomer of this sialylated A structure. Sialylated Lewis A is an isomer of X and vice versa. Here in Lewis A, the linkage between the fucose and the N-acetylglucosamine is alpha-1,4 in A versus 1,3 in Lewis X. It's beta-1,3 in A here between the galactose and the N-acetylglucosamine in A, but it's beta-1,4 in X. I don't want to go into too much detail except to let you know that sialylated Lewis X is an important molecule in itself. It's known as CA19-9, the most important marker when someone has a mucinous tumor like a colorectal cancer or pancreatic cancer or even lung cancer or cholangiocarcinomas. We look for the expression of CA19-9, sialylated Lewis X in the bloodstream, because it's shed from the cancer and it can actually be used as a marker of the total load of cancer in the patient. So when we give chemotherapy and we're asking the question, did we cytoreduce or kill the bulk of the tumor? We're going to survey the levels of this molecule, sialylated Lewis A, which is shed off the surface of the cancer cells. And therefore we can enumerate in a prospective way, prognostic way, whether we're achieving an endpoint of treatment for the cancer. Now, having said that, why do cancer cells exploit expression of sialylated Lewis A? because it allows them to metastasize. So all cancers that come from mucinous tissues metastasize on the basis of expression of sialylated Lewis A, and it's looking for E-selectin. So when breast cancer metastasizes to the bone marrow, it does so because it expresses sialylated Lewis A. When lung cancer metastasizes to the bone marrow, it does so because it expresses sialylated Lewis A. So here's a picture of me about, I want to say about 24 years ago, in the operating rooms of the Massachusetts General Hospital performing a bone marrow harvest. And then over here, my colleague is setting up a filter set, and we'll be passing 11 gauge needles into the posterior superior iliac crest of this donor. And Dr. Murakami is doing this, 3 or 4 times a month now. And this material will be passed through a filter set in order to allow it to filter out bony spicules and fat, which would otherwise cause an embolism in the recipient. And we'll collect a certain number of cells, enumerate them based on the recipient size, and therefore transfer this collection into a recipient. Now, at the outset of my career as a bone marrow transplant physician in the 1980s, we had no clue whatsoever. I like to tell people we had no sweet clue, pun on the word sweet, as to how the hematopoietic stem cells and progenitor cells that we were recovering from the marrow were actually getting into the recipient marrow. Because when you do a bone marrow transplant, you don't inject the cells obtained from the marrow directly into the marrow recipient. That was tried, been there, done that in the 1950s in dog models. And it was actually paradoxically found, I really should say, that if you injected the cells directly into the marrow, you actually had worse hematopoietic recovery. That is, blood cell formation was actually dampened if you tried to inject the cells directly into the marrow. But if you injected it into the bloodstream of the recipient, the blood formation was much more efficient. And that was a funny observation, because you're injecting it right into the site where the cells are being made, but you're actually not getting an outcome equivalent to just injecting it in the bloodstream. Well, it took decades to figure out that when you're injecting it into the bone marrow, you're actually disrupting the bone marrow microenvironment. The microarchitecture of the bone marrow is actually being destroyed by the pressure of injecting a needle in solute and solvents into the marrow. So the cells in the suspension are actually causing destruction of the marrow architecture. But the cell then has some innate capacity to enter the marrow. What and how does it do that was the question of my laboratory. What is the human bone marrow homing receptor? So from 1987, at the outset of my career of bone marrow transplant, into 2000, this was a central question of my investigative effort. I wanted to identify the bone marrow homing receptor, the molecule that pilots, literally, the migration of human hematopoietic stem cells into the marrow, especially since that's what we're doing in bone marrow transplant. So I alluded to the role of E-selectin in tethering cells in hemodynamic flow onto target tissue endothelium. Well, E-selectin is constitutively expressed in bone marrow microvascular endothelial cells. And my laboratory was one of the laboratories that helped make that seminal observation. So therefore, a cell in blood flow that has an E-selectin ligand is going to have an advantage in gaining access to marrow environment. And the cells will then begin to sort of roll along the E-selectin. It'll start tasting chemokines. In this case, the chemokine CXCL12, otherwise known as SDF1 alpha, which is very highly expressed within the bone marrow microenvironment. And the hematopoietic stem cell has a receptor for this molecule, receptor called CXCR4. When that receptor engages this molecule, it upregulates integrin adhesiveness, another class of adhesion molecules, integrins. And there's a molecule on the endothelial surface called vascular cell adhesion molecule 1, which allows, through VLA4 on the surface of the cell, for the cell to plaster down on the microvessel of the bone marrow and then make its way through into the parenchyma. This is transmigration. And now we're talking about parenchymal distribution down here. All right, so what is the most important molecule that engages E-selectin, the linchpin of this process at the beginning, at the outset? It's this molecule H-cell. H-cell is a glycoform, that is a carbohydrate decorated form of CD44. CD44 is a molecule, a protein found on essentially every cell of the body. But uniquely and most prominently, hematopoietic stem cells display a carbohydrate modified form of CD44 that is heavily decorated with sialylated Lewis X. And this thing is the most potent E-selectin ligand. It's actually also the most potent L-selectin ligand. So it's a L and E-selectin ligand, and it's on hematopoietic, human hematopoietic cells, not on the mouse hematopoietic cell. Mice do not express 8-cell because its CD40 form is not decorated with sialylated Lewis X, only in the human. So if our laboratory would have been trying to find the bone marrow homing receptor and using a murine model, we would have never, ever made this observation. So it's important to recognize that CD44 should not be called an E-selectin ligand or an L-selectin ligand, because CD44 is the backbone protein. Only if that backbone protein contains S-lex does it become an E-selectin ligand or an L-selectin ligand. And therefore, the molecule should be called 8-cell when we're talking about the specialized glycovariant of CD44. So if you put this whole thing together at the outset, a hematopoietic stem cell, this is the membrane of the hematopoietic stem cell, will be engaging its E-selectin ligands, and it expresses 3 of them: 8-cell, the CLA glycoform of PSGO1, and the E-selectin binding glycoform of CD43, CD43E, and all of these are looking, literally, for E-selectin. The second they find E-selectin, they're going to grab at it. Now, let me tell you something about this grabbing. This is happening under hemodynamic shear conditions. Let me explain to you that a cell in blood flow can move as fast as 150 miles an hour in the highway of blood flow. These interactions will stop and tether that cell within a nanosecond. So if you're talking about a molecular brake, there's nothing more potent as a brake than these E-selectin interactions with its ligands. And if you just think for a second, just for a second, how powerful the brake would have to be able to slow down a car in a nanosecond on an expressway ramp like 826, as I do see people traveling over 100 miles per hour on that road. If they didn't apply a brake to get off the ramp, they would crash and burn. They have to apply a brake. Well, their brakes better be pretty potent. Imagine the brake that occurs when E-selectin and ligands get together at the outset of this recruitment of a hematopoietic stem cell into the marrow. It's pretty impressive. So here's CXCR4 that binds to the chemokine CXCL12. Here's the VLA4 integrin that gets upregulated in its adhesive capability by G protein coupled activation, which allows it to bind to VCAM, and then the cell transmigrates. Bingo. So now we've talked about E-selectin and its important role in recruitment of cells. And interestingly, I haven't talked about how it works to recruit leukocytes, but you can make the connection. This is the principal molecule that recruits all leukocytes at sites of inflammation. Importantly, saves our bodies from infection everywhere at all times, because leukocytes have E-selectin ligands on them. But I want to come down to this point here. E-selectin receptor ligand interactions are critical to hematopoiesis. So here's normal peripheral blood, what it appears like if you have normal blood. This is what you look like if you have a leukemia. So in other words, this is the normal ratio of a neutrophil, a granulocyte, to red cells in a normal smear. Look at the incredible number of these nucleated cells in the same density of blood. This is what kills you. The fact that you crowd out normal elements and start making these guys. And what am I talking about, crowding them out? Well, this is what the normal bone marrow looks like, with a variety of different cell types differentiating in this matrix. This is what a leukemic marrow looks like. Nothing normal in there. There's very little erythroid progenitors in this picture here. Very few normal appearing granulocytic progenitors, maybe a couple of here and here and here. But the rest of this is blasts. These are leukemic blasts. These are large nucleated cells with a nucleus that normally contains no nucleoli. Look at the number of nucleoli that you see here in these nuclei. These are nuclei, these little pale things, these little pale circle things within the nucleus. This is not good. There's no way you can survive because you're not making white cells, you're not making platelets, and you're not making red cells. Your body's going to die, if not from infection, then from bleeding, and if not from either one of the two, from anemia. So therefore, we got to stop this process and we got to cure this disease. Now, what's happening here is under normal hematopoietic development, E-selectin drives hematopoietic differentiation and also confers critical biology on the hematopoietic stem cell, allowing the hematopoietic stem cell to go into cycle and then either propagate itself or differentiate. But on the other hand, E-selectin also drives the abnormal leukemia niche, which allows the leukemia stem cell, the LSC here, to get a toehold and actually be resistant to chemotherapy. So this is what we're after. And in this review article, they highlight CD44 as being the principal E-selectin ligand. They shouldn't have called it CD44. They should have called it H-cell. Okay. So here's a review article that's about to be published by myself and Evan Alice, as you know, an MD PhD student, the first MD PhD student in the history of HWCOM. And I will distribute this, or Evan can distribute it to anybody who's interested. But in this article, we wanted to identify the role of E-selectin receptor ligand interactions in native hematopoiesis versus that which happens in leukemogenesis. And critically, E-selectin binding in native hematopoiesis increases cell cycling of the native hematopoietic stem cell. So it's a good thing. It's actually fueling hematopoietic stem cell biology. That's a good thing. All right, from the standpoint of the stem cell itself. But if you come over here to what's happening with E-selectin and the leukemic stem cell, not the propagating leukemic cell that has a very high turnover rate and is cycling greatly. I'm talking about the one that's resistant to chemotherapy because it's not cycling. E-selectin fuels the dormancy of the leukemic stem cell, where here it increases the cycling, here it increases the dormancy of the leukemic stem cell. That's important. So people are creating little mimetic molecules of sialylated Lewis X to disrupt the niche in leukemia to try to get access to these leukemic stem cells, is what we call leukemic initiating cells or leukemic repopulating cell, either LIC or LRC. You'll see the same name for the same cell, or it's different names for the same cell. Here, E-selectin antagonists, little mimetics that look like SLEX, will enhance the LIC/LRC cell cycling, increasing their chemosensitivity. Whereas when you're giving these things and you're trying to treat somebody with chemotherapy, in the case of the hematopoietic stem cell, the antagonism will actually lead to quiescence of the hematopoietic stem cell. So it will protect the normal hematopoietic stem cell, but it'll induce these little mimetic molecules will interrupt the E-selectin receptor ligand interactions on a leukemic stem cell, allowing it to go into cycle and therefore be amenable to destruction by cycle-specific agents or a variety of different means that we can get at cells that are repopulating or dividing rapidly. So we have a grant here at FIU to study this, and it's part of this program out of the National Cancer Institute. And we have a variety of different aims. We're identifying the ligands of acute leukemic cells. We're trying to identify the transcriptome of genes programming the expression of sialylated Lewis X on acute leukemia cells. And then we're defining how these E-selectin ligands impact leukemia cell survival. This is work that's ongoing in our laboratories currently. So one way to get at this disruption of E-selectin receptor ligand interactions is to create mimetics of sialylated Lewis X. But we've taken an orthogonal approach. I'm going to share that with you in the last few slides of the talk. So here's our structure, sialylated Lewis X. Again, neuraminic acid is also called sialic acid. So you see the neuraminic acid here, alpha-2,3 linked to lactose, beta-1,4 linked to N-acetylglucosamine, alpha-1,3 fucose sitting right here. Whenever a glycan is constructed within the Golgi, it's an assembly line process where the glycosyltransferases function in sequence, just like an assembly line in an automobile factory. You can't put the chassis onto the motor, until you have all those parts of the motor and the wheels in place. And then once you have a chassis, you have to put on the glass. So you can't put on glass when there's no chassis, and so on and so forth. Well, the glycosyltransferases are looking for their acceptors, the specific precursor molecule that they know is where they need to act, and they do it in a sequence. So when you construct, or the cell, because you are constructing it, when your cells are constructing sialylated Lewis X, they're first looking for this galactose N-acetylglucosamine linkage, this lactosamine backbone. They'll then lay on a sialic acid on that. But if there's no galactose there, they'll first put on a galactose. So here's a an empty N-acetylglucosamine doesn't have a galactose, the galactosyltransferase senses that, it puts on a galactose. Then the sialotransferase is seeing that there's plenty of opportunities now with at least 2 more galactoses placed on top of N-acetylglucosamine to land the sialic acid on there. And that's how it works in a sequence, okay? So if we take this apart, we know all of these enzymes. There's redundancies in the galactosyltransferase. I'm sorry, there's not a redundancy in the galactosyltransferase. There's only one that'll put a galactose on top of an N-acetylglucosamine, and that's beta-4 galactosyltransferase 1. But there's redundancy in the sialotransferases. There are 3 different sialotransferases that can place a sialic acid here on top of this backbone. And then there are 4 principal ones and a 5 one, which is another one, which is called fucosyltransferase 4, which is a little bit anemic. They can place a fucose in alpha-1,3 linkage on this N-acetylglucosamine, completing the tetrasaccharide, the 4 monosaccharide linkage units like shown, that makes sialylated Lewis X. Now, why is that important? We want to disrupt production of sialylated Lewis X. So we reasoned that we're going to want to disrupt this terminal sequence right here because it's the easiest one to get to. It's at the very end of the chain. So I don't want to dive into too much detail here, but a lactosamine has a lot of choices on where it can go and how it can go, and not the least of which is, incidentally, your blood group antigens. Your blood group antigens, your type A, well, functionally your type O, and your type A and type B are all based on a lactosamine backbone, which is terminally decorated by different sugars to create the O phenotype, then the A and the B phenotype. I'm not going into detail on that right now, but the bottom line is that there's a lot of places for a galactosamine. There's a lot of places for a lactosamine to be modified. But if it goes down the path of getting a sialic acid and coming here, we want to interrogate this. We want to stop this. And we know unequivocally that leukemia cells are laden with sialylated Lewis X. Now, some of you might ask, well, do they have sialylated Lewis A? No, they do not, because they don't express the capacity to make a beta-1,3 linkage between the galactose and the N-acetylglucosamine. So remember in the prior slide, I said there's one beta-4 galactosyltransferase that can make this, and that's the one that's on hematopoietic stem cells. They don't have the beta-3 galactosyltransferase that would make the otherwise known as a type 1 lactosamine backbone, where this is a beta-1,3. We don't have that in hematopoietic cells. We only have beta-4, GALT1. So if we see a naked N-acetylglucosamine at the terminus of a sugar, and we modify with a galactose, it's going to be in a beta-1,4 linkage. Once that's there, it locks in that the only place that's open is the 3 position on this acceptor monosaccharide. Therefore, it has to be an alpha-1,3 linkage. I can go into greater detail for those of you that are interested in that kind of detail. But the bottom line is hematopoietic cells only make sialylated Lewis X. So these are the terminal lactosamine choices of hematopoietic cells. And then just want to comment on the production in critical terms of sialylated Lewis X. All right, so here we are on this limb of this production schema. Incidentally, this right here is blood group O. And if this galactose gets modified by another galactose, that's blood group B. If this galactose gets modified by an N-acetylgalactosamine, that's blood group A. But this core, alpha-1,2 fucosylated lactosamine, is the H antigen, otherwise blood group O. So let's go now to where this is going to the terminology and why this is so critical. So we have the alpha-1,3 fucosylation, the terminal step in S-Lex production, and we want to get into this and try to stop it. So that's precisely what we've been doing in the laboratory in collaboration with Dr. Barbara Riccici, who is a world-class glycochemist, creating structures that look like donor fucoses to interrupt the fucosyltransferases that would otherwise make sialylated Lewis X on hematopoietic cells. Now, when we're doing this, we know that if we succeed in so making this compound, we're going to disrupt normal hematopoiesis. Yes, we may be disrupting normal hematopoiesis, but we want to time this at precisely those times where we're giving the chemotherapy that is going to kill the most rapidly producing cells And if we disrupt the content of sialylated Lewis X on a hematopoietic stem cell, it'll basically allow it to go dormant. But at the same time, it'll fuel the increased cycling of the leukemic stem cell. So this is a paper that was recently produced, and Kyle, at the beginning of our meeting today, talked about, we just got our first citation of this report, and incidentally, a patent has been issued for the construction and the utility of these fucosyltransferases, these inhibitors of fucosyltransferases that we've created in the laboratory by exploiting fucose mimetics. So with that, this is what we're aiming to accomplish: use of fucosyltransferase inhibitors to disrupt E-selectin receptor ligand interactions, which are critical to leukemogenesis. And here's a leukemic blast. And we recognize that when this cell is binding to E-selectin, it has decreased apoptosis, decreased sensitivity to chemotherapy. But if we disrupt this, we'll increase its capacity for chemosensitivity and increase its likelihood of death itself. Now I want to end this talk by just bringing to your attention the excitement in the glycobiology community that one of our own got a Nobel Prize recently, because it's very difficult for us to talk this talk. And many of you listen to this, get excited because you hear about its applicability, but to understand its applicability, you have to understand the underlying chemistry and the specificity of these linkages. And that's a daunting task. It's challenging, and a lot of people run away from it. But if you can stay in it, it is the most fascinating field of biomedical science you can possibly imagine. It is so incredibly fascinating. But many years ago, I was telling my wife, Beth, how important glycobiology was to my career. And she said, well, guess what? I got this thing in the mail. And actually, I didn't realize it, but glycobiology is critical to all these skin products that I'm using on my skin. And I said, really? Well, people don't realize hyaluronic acid is a simple disaccharide polymer. Hyaluronic acid is a glycan. But of course, glycans are in the extracellular matrix. They're holding us together. The proteoglycans, the molecules that are called proteoglycans, have glycan decorations, which are part of the ability to cross-link and keep our tissues together. So sugars are really critical to all biologic events, importantly, the construction of connective tissue. But she said, look, I got this just now from Saks Fifth Avenue. It was a flyer. And it was to visit the Yves Saint Laurent counter in the cosmetics section at your nearest Saks Fifth Avenue to receive a complimentary sample of Forever Youth Liberator. Pretty interesting. So this caught my attention when she brought it to my attention. Yves Saint Laurent Forever Youth Liberator, 100 years of research, 7 Nobel Prizes, now 8, but only 8? Of all the Nobel Prizes ever been given out, only 8 have been given out to somebody who knew something about sugar chemistry? That's ridiculous. That's because the field is so difficult to get your hands around that most people can't appreciate it. So we've had 7 Nobel Prizes, now 8. We're underrepresented, to be honest with you. But here he says, only 1 discovery, glycans. Really? Only 1 discovery? 1 discovery. That's ridiculous. But that's what this little advertising blurb says, okay? But now it gets really interesting. Here's the backside of this. Inspired by glycobiology, the science of the future, after DNA and stem cells, glycobiology is recognized as one of the breakthrough sciences of the future, awarded by 7 Nobel Prizes, now 8. Glycobiology provides deeper understanding of the body's biological mechanisms. Glycobiology is the study of glycans, molecules which are naturally present in the skin. Not just the skin, my friends. Glycans are responsible for directing communication between cells, ensuring that they send and receive the messages required for optimal performance. That's an understatement. Without glycans, cellular communication breaks down and the body cannot properly function. Hear, hear. So here's the major discovery in anti-aging. Yves Saint Laurent introduces Glycan Active. This stuff is actually on the counters right now, and it costs over $200 for this little vial. So get that. A patented complex of glycans which imitates the actions of glycans, helping to restore your skin's youthful beauty. So with all this, I say, look at that, guys. Yves Saint Laurent is a translational glycobiologist. So we've got this incredible opportunity here at Florida International University. Our colleagues don't even understand it. Nobody really gets it. We get it, and we are going to continue the efforts to improve human condition through biomedical science that will offer curative intent therapeutics grounded in glycobiology. I want to thank the members of my laboratory that have been involved in this work. Kyle Martin, lab manager, Evan Alice, the MD PhD students doing fantastic work in understanding E-selectin ligands and leukemogenesis. Dr. Kevin Chandler, who's premier glycochemist and also now increasingly glycobiologist and expert in glycoproteomics. And Dr. Agatha Steenackers, who was in our laboratory and just recently left to be a member of a laboratory group at the Medical College of Wisconsin, the Blood Center at the Medical College of Wisconsin. I've got very close colleagues that are collaborating with me at the Dana-Farber Cancer Institute. I want to highlight Dr. Mark Murakami, who's on this call today, Dr. Rich Stone, who's the head of leukemia service at the Dana-Farber Cancer Institute, Dr. Konstantin Mitsiadis, an expert in the hematopoietic microenvironment and an expert in multiple myeloma, and Jeff Sorrell, lab manager, and Dr. Mitsiadis' laboratory. And lastly, Dr. Barbara Riccici, who has helped us in developing these inhibitors of transferases, which we believe will be critical to saving the lives of many patients. Thank you very much for your attention. I'll take some questions.
I have a question, if that's okay.
Yes, of course.
Yeah, great talk, Robert. There were a couple of questions I had. You said that some of the sialylated Lewis A and X moieties could be detected in the circulation. What is the mechanism of shedding? Is that cell death and it's basically debris, or is it actually? And what is actually circulating? Is it the Lewis A or Lewis X in isolation, or it's still conjugated to other parts of the carbohydrates chain?
Absolutely. Yeah. It's conjugated to both lipid and proteins. So it really is using an antibody detecting only the carbohydrate motif itself, but it is still is a glycoconjugate on a lipid or protein backbone. Those molecules are shed all the time from cells, and cells that are rapidly turning over tend to model their membrane more readily than native cells or healthy cells. So you have the added advantage that those things are naturally turning over. There are certain molecules that are shed by virtue of proteases that will hit certain classes of molecules, and therefore there's a mechanism that is enzymatic cleavage. But for the most part, it's just a natural turnover of the membrane and the removal of certain molecular components on a regular basis.
I see. And do you know if it's possible to conjugate to the glycomimetics? Can you conjugate those to other targeting moieties? In the case of that leukemia trial that you were describing, you were very elegantly proposing to exploit the benign malignant, the distinctions in biology between the benign and the malignant cells, where in fact you want to target both, but in other applications, if you wanted to target one particular cell population, there may be an advantage if you could somehow anatomically or cytologically target the mimetic. Is that
yes, there have been in the outset of this field, there were interest in liposomes labeled or laden with sialylated Lewis X. What's interesting about the glycomimetic compound is literally a small analog, a small molecule analog that has a very high or very low KD for the binding to E-selectin. So therefore, you can give something that looks like a tetrasaccharide. And that's exactly what GMI 1271 and GMI 1273 are. They're mimetics that look almost like sialylated Lewis X. I believe that we can converge a mimetic with a fucosyl transferase inhibitor and double the potency, if not logarithmic increase the potency of disruption of E-selectin receptor ligand interactions in the therapeutic algorithm for leukemia. And so we do believe that there's a chance, because they're orthogonal approaches, they can be used concurrently. And yes, there are approaches probably still ongoing to provide multivalency of sialylated Lewis X to possibly increase its capacity to disrupt with higher efficiency. Does that answer the question?
Yes, fantastic. Thank you.
You bet. Any other questions? Well, Thank you all so much for your attention today. I hope everybody has a great weekend. I think by hearing this conversation or discussion today, people should understand what translational glycobiology is. By all means, take a look at the review article that I highlighted by Dr. Bertozzi. It's a really wonderful review of why glycobiology is important. But remember, Dr. Bertozzi is a PhD. She's never felt the pain of actually seeing a patient die. And I want to tell you that what got me launched on this was an observation that I made, unfortunately, in the late 1980s, when I did perform a bone marrow harvest, and I collected a sufficient number of nucleated cells to have perceived that the outcome would be great, and the person never started making blood cells. We call that failure of engraftment. And at the moment that I realized that I was the reason for the death of the patient, because this person had aplastic anemia and would have been able to survive probably 5-10 years whimpering along with aplastic anemia, but we said, we're going to save your life. We're going to replace your diseased marrow with healthy marrow, and therefore you're going to be able to make all kinds of blood cells again that you can't make now. Instead, we ablated the marrow in order to make quote unquote space for the donor hematopoietic elements. And then the donor hematopoietic elements never took off and the patient died. And I literally was crushed because it wasn't a 50-year-old. At that time, we only did bone marrow transplants for patients under the age of 35. The person was 16 years old. So if you can think, those of you that are in medicine, how it feels to lose a patient, how it feels to prepare a patient for a procedure which is supposed to be life-saving, and to actually perform the harvest, knowing full well that you're doing everything in your power to do that procedurally as well as anyone could possibly do it, feeling good that you've collected enough cells, injecting those cells in the marrow, and then finding that the cells never created the kind of blood cells necessary to sustain life. And your hands are doing that. That gets you in the lab for the rest of your life, my friends. End of story. Have a great day. Be safe. Talk soon.